So it's my distinct pleasure to introduce our next speaker, who is Dr. Vandana Gupta, who works on neuromuscular disorders with a particular focus on the molecular mechanisms underlying genetic muscle diseases, gene discovery, and therapeutic development. Her research integrates genetics, developmental biology, and translational approaches to uncover disease mechanisms and develop targeted therapies for rare neuromuscular disorders. Vandana currently oversees the TANGO2 Research Foundation Scientific Advisory Board, so please help me in welcoming Dr. Vandana Gupta.
Thanks, Donna, for the kind introduction, and I would like to thank the organizers for the invitation to the meeting and an opportunity to share our work on TANGO2 therapy development. TANGO2 is a recessive disorder where we have mutation in both the alleles in TANGO2, resulting in loss of protein function. So either the patients have very low amount of TANGO2 protein or the protein is not made. Disease is clinically and genetically very heterogeneous. The disease onset can be seen in the at birth, early childhood, or in some cases even in the adult patients. So that adds to the complexity of any therapeutic development and also when to give the therapy to these patients. We have a genetic disorder when we don't make TANGO2 protein. And on top of that, there are external factors that can complicate the disease. The disease onset can be due to in some patient in the absence of any kind of external factors, or it can also be due to any of the external factors such as infection, dietary, fasting, exertion, or maybe some other unknown factors that we are still not very clear about. And this point is important to remember because any of the therapy that we develop for this TANGO2 patient is going to get affected by the kind of trigger that cause metabolic crisis in this patient, or kind of stressor that can lead to disease in these patients. And I'm going to show an example later why this point is important. And of course, there's an unmet need to develop therapies for TANGO2 deficiency disorders. Between B5 has shown excellent result in prevention of metabolic crisis, but we still see a lot of patients in which we need to have additional therapies to reverse a disease or prevent complete onset of the disease. So when we talk about therapeutic options in recessive genetic diseases, one of the most attractive strategy is the gene therapy. We have a missing gene. Now we put the missing gene back in the patient body by the use of adeno-associated viruses, that has been shown to improve the disease in a number of other genetic disorders, and I'm going to show some example. So we put the missing gene back, the functional copy of the gene makes a protein, and we rescue the function of the gene. And this therapy is really attractive because this is mutation agnostic. So a patient can have any mutation throughout the TANGO2 gene, whether it's a missense mutation, with a loss of function mutation. This therapy can be given to any patient independent of the kind of mutations they have. Another very common approach these days is gene editing, where we can go and fix the specific mutation a patient carries in the DNA. And that using CRISPR-Cas9 approach, and now these patients can make the functional TANGO2 protein. This is very promising and has shown to be effective in some cases, but the big problem with this approach is for every patient, we need to develop one therapy. So every patient with a mutation, we need one therapy. And taking a single therapy to clinic can take years and so much of work. And now if we start to do this kind of therapy for every single patient, there'll be about 200 therapies for TANGO2 patients that will never make it to the clinic. So something which is not really practical for these kind of disease. Third option that we are considering is small molecules, where we can screen a number of small molecules using either cellular or animal models of TANGO2 deficiency. And these molecules may not correct TANGO2 deficiency as such. They may not be able to make TANGO2 protein. But what they can do is, if a patient is making very small amount of TANGO2 protein, some of this compound may be able to stabilize that protein and stabilize the disease progression. Or in some cases, they may target the downstream pathway in which, let's say, TANGO2 is involved, and they can rescue the function by rescuing that pathway. And finally, we can also, in some diseases, gene expression modulation has shown great promise. For example, splicing, we can remove a certain part of the protein, and the remaining protein is smaller than the normal protein, but is still able to function well. But this approach, TANGO2 protein is already so small, this approach is not going to work very well in TANGO2 deficiency. So my group is working on two approaches from this. One is a gene therapy, and one is a small molecule-based discovery using zebrafish model. And I'm just going to talk a little bit about both of those. So in the gene therapy is really attractive to us because in the last few years, a number of gene therapies for all different kinds of genetic disorders have been approved by a number of different companies. And one of the most attractive examples for us is spinal muscular atrophy, because we work on neuromuscular diseases. These are the babies that are born with very weak muscle. They have really loss of neuromuscular function. And since the gene therapy has come now, these babies are not only surviving well into their late childhood, but also the motor function is significantly improved. So this is a great promise to bring the therapy, gene therapy, for many of the diseases. And now in the recent years, we have examples where the therapy has moved from the labs directly to the clinic with a number of small group of patients that we can see for a number of different kind of disorders, with patients as low as 3, in some cases 1 patient. But what this work has done, this has also made the basis for developing this therapy or extending the therapies for a larger group of patients. And one of the examples is spastic paraplegia type 50. This patient had a genetic mutation at birth, and the gene therapy was developed for 1 patient a few years back. Within 3 years, now almost 40 patients throughout the world are being treated with this kind of gene therapy. So both, whether it is developed by a company or whether we are developing in the lab, has great potential to take the therapy to the clinic. So again, coming back to now specifically gene therapy for TANGO2 deficiency. Why do we want to develop a gene therapy when there are some other therapies in the pipeline? One of the reasons is TANGO2 gene is small. It's a monogenic, simple loss of function. So we can put the gene back into the cells and restore the function of TANGO2 protein. And also with a single dose, we can correct the long-term correction. Like we don't have to give the patient compound or a therapy every day, every week, every month. With a single dose, hopefully we can correct the disease for the rest of their life. Of course, there are challenges for gene therapy like any other therapy, which is like which target tissue is going to be most beneficial with the therapy, dosing, and the safety. So what we have done is we have started developing AAV-mediated gene therapy in mice. And I'm not going to show a lot of data from TANGO2 mice, but TANGO2 mice don't have a phenotype. That's one of the biggest challenges we have. Then how do we see the effect of therapy? So right now we are modeling all the clinical stressors that we have seen in the patients in this mice to be able to see which is the most effective stressor that can cause a phenotype in this mice, and then we can study the effect of this therapy in this mice. And again, TANGO2 is expressed in most of the cells in the body, so it is not as simple as some of the other therapies we have developed. And I'm going to show you an example. Which promoter do we use, like which tissue type we want to express to prevent the most effectiveness and least amount of toxicity. And finally, as we express AAV TANGO2 in this mice, we want to evaluate not only the efficacy of this disease, but also the biodistribution and safety. And so I just want to show you next few slides of a small example of a therapy that we have developed, which we are taking into the clinic in next few months. One of the diseases we are studying results in muscle phenotype, where the babies have really weak muscle and they die within few months after birth. So we created a mouse model, and these mice also have very weak muscle, similar to these babies, like 6 days after birth. And most of these mice die within 1 to 2 weeks after birth. So we have started looking at the muscle function in this mice. These are 4 days old mice, which we put on the side, and they get back on all their 4 feet. These are the control mice. And on the right are the mutant mice. So they have such weak muscle, they're not able to get back on all of their 4 feet for a very long time. So we were dealing with such a severe disease here. And for this disorder, we developed a gene therapy where we put back the normal copy of this KLHL40, which was a missing gene, using AAV9 serotype. And what we see right now, our mice are about 8 months old. And the mice that die at 1 week of age, similar to the patient, are surviving to the longest time we have studied so far. So we are going to see how far these mice can live. And this is an example. This mice die so early without treatment, but with treatment, most of these mice are surviving. And then they eventually gain weight, very similar to the control mice. So we can look at a number of different factors when we treat the mice or any other animal with any therapy. And here's an example where we looked at how fast they can move. 30 days after the therapy, they restore the muscle function to 50% of the capability to the control. And after 3 months of therapy, they are as normal as the control mice. And this is an example I like to show. So one of these mice is normal. Another mice would have died at 1 week of age without therapy. And even, like, we cannot tell which one is which. So as we develop this therapy, we also are very careful about the safety profile, because we do not want to just develop a therapy and take it to the clinic without knowing exactly what are the potential safety issues that can happen, or learn that by clinical trial where we see adverse event in the patients. So we have looked at the biodistribution profile, for example. Is the protein is expressed in the tissue where it is supposed to express? In this case, our protein is expressed in different skeletal muscle type and to some extent in heart. And even in the treated mice, we see the similar expression profile into different tissues. Here, this is a skeletal muscle section, and the green color is the protein of our interest. And we see in the treated mice also, it is expressed in a very similar pattern. It is present in the normal mice. And again, we go and do scan all the different peripheral tissue just to make sure that there's no phenotype, because pathologically we can see changes even at the level of, especially in the liver. A lot of gene therapy studies have been halted in the past because of liver toxicity. So we are always concerned to make sure that there are no obvious changes in the liver. We are now doing a number of molecular study by RNA-seq and proteomics to see that maybe we are not able to see any big toxicity in these mice, but can we find some small molecule signature that can tell us that there are going to be safety issues. So I just wanted to show those slides to give an example of the similar work that we are doing in the TANGO2 mice, and hopefully in next few months we'll have data on TANGO2 mice on our gene therapy work. So while we are doing the gene therapy work, another approach, as I mentioned, we are taking is a small molecule-based screening in zebrafish. And one of the reasons we are doing is that we understand that not every therapy is going to be safe or possible for every single patient. So it is always better to have a number of different therapies that can be given to different patients on the basis of their disease symptom, disease severity. So a few years back, we developed a zebrafish model of TANGO2 deficiency. This fish were completely normal. They swam normal. We didn't see any phenotype in this fish, even though this fish did not make any TANGO2 protein. So this was very surprising to us. Like, we tried to look at all different angles under the microscope to see if we could find any small thing, but for months, we could not find anything. So what we did was, before we went ahead, we decided to look at the muscle structure, because when the metabolic crisis happens, skeletal muscle breakdown or rhabdomyolysis is one of the most prominent features. And because skeletal muscle is 80% of our body weight, we wanted to see, do we have anything happening before any crisis happens in this animal model. So this is a normal skeletal muscle, actin-myosin organization. And these are the mutant zebrafish. And what we saw was that compared to the control, the mitochondria, this is a normal mitochondria, and this is a mutant mitochondria. This was very abnormal and not well developed. And it's not that all the mitochondria were bad, only a small percentage of mitochondria in this fish were bad compared to the control. So this was very fascinating. This was the first evidence we saw that in the absence of TANGO2, they are actually pathological changes in the body that are affecting mitochondria. So one of the thought we had was that what if we do drug screening using small molecules that can affect mitochondrial function? And in order to do the drug screening, we needed a couple of different assays. So one of the things that we modeled in the zebrafish is fasting, because fasting is shown to cause metabolic crisis in the patient. And in this case, these are normal zebrafish. These are our TANGO2 zebrafish at the baseline condition. And then we did not provide them external food for a couple of days. While wild-type fish, they are more robust. They were perfectly fine. And what we are looking at the swimming ability or the total distance moved. This wild-type fish were perfectly fine, but the TANGO2 mutant zebrafish, they just crashed. They did not move at all. So we thought this is one of the very robust assay to look at the effect of different compounds on their ability to restore this swimming ability back to normal. And the second assay we did was exertion. So in this case, we increase the viscosity of the swimming water. So now the fish have to put more pressure or more force in swimming in the same thing, which causes a stress in the skeletal muscle. And these are the example on the top are the normal skeletal muscle staining by actin staining. These are the zebrafish, which are TANGO2. We see they're not as perfect as normal fish, but still the muscle overall does not look very bad. And these are the example when they swim in this viscous water, their skeletal muscle just completely breaks down, that we quantified by the swimming ability here before adding the methylcellulose or before increasing the viscosity, they just swim normally. But when we increase the viscosity, they just crash. And we have looked at it into different zebrafish models of TANGO2 deficiency. So then what we did next was we took this zebrafish and got this library of compound that has about 998 compounds. We added one compound to each well of a multi-well dish that has a bunch of embryos with TANGO2 deficiency. And then we tested two stressors. One is fasting and one is overexertion to see which of these condition and these compounds. So one of the questions we had is that, are we going to identify the same compound in both the condition? Is the discovery of the compound is going to be based on TANGO2 deficiency? Or is it that this different stressor interact with TANGO2 deficiency and result in different kind of effect, and we will identify different kind of effects. So while we were doing that in a collaboration in a parallel screen, Sam's lab also did the similar screen in C. elegans worm using oxidative stress as one of the stress condition. So our screening identified 35 compounds that we identified in the fasting condition. We identified 5 compounds in the exertion conditions and 5 compounds that in fasting and exertion condition. And I think one thing I wanted to mention was that we provided these compounds very early during development, before we see any of the mitochondrial defects that I showed you in the muscle. So the disease is not yet established. We know this fish have TANGO2 deficiency, but we still do not see any changes in the mitochondria or anything early onset. So at this point now, we are looking at testing this compound and reversing the disease onset. Can we reverse a disease when it is already there? And how late can we reverse the disease in these conditions? So we're doing a number of different things, checking the disease, these compounds at different dosages, and also looking at the downstream pathway. So not all the compounds in this library are FDA approved. And one thing we can do is that any compound that we identify, if that is not FDA approved, there are many similar compounds that are FDA approved for some other disorder. And right now, I do not want to say too much about these compounds because we still need to do more work. But some of the compounds that we have identified and are not FDA approved have other compounds that are FDA approved targeting the same pathway. So we want to now bring those FDA approved compounds also and test whether they can prevent the disease onset and whether they can also reverse the disease back to the normal state. And finally, again, most of the studies we are doing in zebrafish, which are vertebrate model, but we still want to test in patient cell lines and mouse model that are more relevant to the human patient before taking any therapy to the clinic. So just to conclude, we are developing therapies for TDD using multiple approaches, in particular gene therapy and small molecules. And as I mentioned earlier, finally the optimum therapy may vary by disease severity in individual patient, the gene affected tissue, not gene, the mutation affected tissue, and the patient. And in some cases, even integrated approaches in combination may provide great benefit than any of the single approach. So with that, I would like to thank all my lab members, especially Grace, who did most of this work with the help of many other members of the lab, Sam and Sarah from University of Rochester, Baylor School of Medicine, Christina, Na, and Lily, who sent us a TANGO2 mice, and all the funding agencies, especially TANGO2 Research Foundation. Thank you.
Hi, thank you. Great talk. You talked about doing fasting and overexertion in the zebrafish for the phenotype. Have you tried that in the mice yet yourselves?
So we have, and we are analyzing the data from those studies. Not the exertion yet, but fasting, as well as we are trying high-fat diet, because the group in Australia published that high-fat diet, they made the phenotype worse in the mice, so we want to see if that works for us as well.
Yeah, I'm still in the process of doing similar things, and I found that overexertion has been the clearest personally. So, but I'm also still working on that.
Okay, thank you.
My name is Reese Moore, and my sister has TANGO2. But with the gene therapy in the mice, were y'all able to find that it was able to correct it in the brain also?
We hope so. To what extent, we are not clear yet, but in gene therapy has been shown to reverse back many of the disease symptoms of the brain in many other disorders as well. Yes.
Thank you, Vandita, for that presentation. Just a quick question on the gene therapy in the mice, just to kind of touch upon the previous question. Are you targeting a particular tissue with this, or are you going, or is it multiple body systems?
So the first promoter that we are using, we are going to target, is going to be ubiquitous. It's going to go into every cell in the body, brain, muscle, heart, everything.
Thank you.